All right, I have several things to say first. All right, I'm an enzymologist. I am not a bioinformatician, and so I see that I'm supposed to be giving a bioinformatics lecture, and I really don't know what that means, okay? I'm an enzymologist who is interested in assigning functions, as you'll see, to uncharacterized enzymes in genome projects because I'm interested in understanding or discovering new chemistry, all right? So we, as I'll tell you, develop these tools. Also, as I told Karsten, I think, I used to think that metals were magnesium, all right, because that was the most complicated or biggest, only metal I had ever worked on. And of course, I come to a conference like this and I realize nobody cares about magnesium. So I'm a little bit out of context with respect to the bioinorganic, but hopefully you'll find the tools that we have developed useful. Okay, so the problem is that that we're all confronting is that it's very easy now to determine the sequences of genomes, and this is a plot of the number of sequences as a function of time in the UniProt database, and this is actually the Tremble database, which is the automatically annotated part of the Tremble database. Or the UniProt database. The UniProt database is maintained by EMBL EBI in Europe. And as you can see, the number is increasing rapidly. This now, as of about a week ago, contains nearly 116 million sequences. UniProt also maintains the SwissProt database, and there are data on this slide. The data are basically buried on the x-axis. The SwissProt database are the sequences in the UniProt database that have been manually curated. That is, someone has gone through the literature and associated a published function with these. Okay, so you can see that there are only about a half a million of these. So only a very small fraction of the sequences in the UniProt database have manually curated functions. Of course, some of these functions can be extended by homology orthology so that you can try to pick out what orthologs might be, proteins with the same function, but it's very difficult to predict what the sequence boundaries are between functions, and so you never know quite how far you can extend homology, okay? So there is a gap here between what we know for sure and what is automatically computationally assigned. And this is a little disconcerting. It's disconcerting to us enzymologists who are interested in discovering new functions. In the past, I've been interested in evolution of enzyme activities within mechanistically diverse enzyme superfamilies, and I've very much wanted to be able to fully identify the functions or identify the functions, complete set of functions for a family so that I might be able to figure out how the functions evolved. But without knowing what the functions are, what the substrates are, or the reactions, you have a problem, okay? So that's how I got into this game. All right, so over the years, I've been involved in several program projects, and also for a while, for 5 years, a very large collaborative project that was supported by NIGMS, and this is the Enzyme Function, was the Enzyme Function Initiative, and actually for a while, Squire was associated with that. And what the Enzyme Function Initiative was trying to do was to devise tools and strategies to enable the assignment of functions to uncharacterized proteins discovered in genome projects. And starting with sequence, we would predict or determine a structure, we would predict reactions computationally, and then experimentalists like me would determine functions. So as part of that, we developed some tools, and when I say we, I really want to give proper credit to Patsy Babbitt, who has been at this meeting before, because Patsy was involved, Patsy was responsible, and I'll give her credit on a slide or two, for developing protein sequence similarity networks. Patsy and I coined the term genomic enzymology a number of years ago, and this is to use the large number of sequences that are available in the protein databases and the large number of genome sequences and genome contexts that are available in genome databases to try to leverage the assignment of functions to uncharacterized enzymes. So the strategy that I want to go through today, and I'm going to give you an overview. Remy Zalat, who is here, and I will be doing tutorials this afternoon, tomorrow, and Monday, and those tutorials will go into the details of what I'm going to be providing as an overview today. So what are the steps of assigning function, or what are the thought processes involved in assigning function? The first is we would like to be able to explore sequence function space in a protein family. We'd like to take a protein family and segregate it into isofunctional clusters. And to do that, we have developed, or Patsy was instrumental in developing, and then we've implemented a web tool for allowing the construction of protein sequence similarity networks, or SSNs. And I'm going to be telling you about the EFI EST tool that enables the generation of these networks. Also, as I've mentioned, it's important, or we believe it's important, to put these isofunctional clusters or to try to identify the genome context of these isofunctional clusters, because in bacteria and in fungi, metabolic pathways are often, not always, encoded by gene clusters or operons. So if you have a protein of unknown function and you can look in the genome context, what you might be able to do is to pick out proteins, enzymes that are catalyzing reactions in a pathway, and maybe you can integrate that information and try to predict the pathway. And I also, if I have time, I want to tell you that we are trying to enable, or we've enabled, a strategy for prioritizing clusters for functional assignment. This is a strategy that was described about a year ago by Emily Balskis and Curtis Huttenauer, called chemically guided functional profiling. And we now have a beta tool which will allow that, which allows that. Okay, so today what I want to talk about first is the EFI EST web tool for generating sequence similarity networks, then the EFI GNT web tool for generating genome neighborhood networks. And even though there is not an iron or a cobalt or a manganese, there is a magnesium or two in this. I want to talk about how we've used an example of using SSNs and GNNs to discover novel metabolic pathways with novel enzymes. And then if I have time, I'll say just a little bit about this chemically guided functional profiling tool that's in development. Okay, so the EFI EST tool. So as I told you, Patsy Babbitt popularized within the enzymology community these protein sequence similarity networks for visualizing sequence function space in entire protein families. So this is a slide that I stole from Patsy. On the right-hand side of the slide is a phylogenetic tree or dendrogram for part of the enolase superfamily that Patsy and I used to collaborate on. And on the left-hand side is a sequence similarity network for the same sequences, the same portion of this, the family, the enolase superfamily. And I always looked at these and said, these are a bunch of twigs and sticks, and I haven't the slightest idea how to interpret them and to separate one from another. But if you look at sequence similarity networks, in the sequence similarity network, each dot or symbol is a sequence, and each sequence is connected to every other sequence in this homologous group of proteins that shares greater than a user-specified level of sequence identity. If you look at this, you can see that there are some clusters, some separate clusters and some emerging clusters, and these clusters are likely to be isofunctional families. So this allows you to look at a large number of sequences and pick out sequences that are likely have the same function. All right. All right. So how do you construct these? Okay. So a dendrogram or a tree is the traditional way of doing this. The evolutionary biologists like to do this, and these are constructed using multiple sequence alignments, and these are computationally extremely intensive, and they are extremely difficult to do for large numbers of sequences. All right? But they are phylogenetically rigorous, and if you're interested in getting phylogenetic relationships, this is what you want to do. A sequence similarity network, as I told you, in a sequence similarity network, each symbol is a sequence, and each sequence is connected by a line or an edge to every other sequence that shares a sequence identity that is greater than a user-specified value. And if you have a very low sequence identity, all of the sequences in a homologous group will be connected in a single cluster. And then as you increase the sequence identity that's required to draw an edge, this hairball, as Patsy would call it, would segregate into into individual clusters. These sequence relationships are calculated by pairwise blasts, all by blasts. And these are much faster to calculate than multiple sequence alignments. So you can calculate sequence similarity networks for hundreds of thousands of sequences. That would be virtually impossible to do for multiple sequence alignments and dendrograms. But even though the trees, dendrograms, are more rigorous and more accurate, usually the results that you get with sequence similarity networks and dendrograms trees are quite similar. So obviously, we prefer to do this. Okay, so I want to just go through what EFI-EST does, what it uses and what it does. And like I said, in the tutorial that we'll be giving, they'll be hands-on. We'll be connecting to a cluster. The EFI bought a cluster, which is at Illinois. So I actually own a cluster, and I think I'm an enzymologist, and I think I like to think about protons and electrons, but I also have to think about nodes and processors, okay? So we will do an online demonstration of this and to show you how to do this. Okay, we use sequences from Uniprot. This is supported by EMBL EBI. Okay, Uniprot has two types of sequences, as I've already told you. There are Swissprot sequences, which are entries which are manually annotated, and then there are the Trumbull sequences that are automatically annotated. This automatic annotation uses BLAST, essentially uses a sequence comparison, and it transfers the annotation for the closest sequence that's already in the database to a new one, and the problem is that is not always right, but that annotation gets in the database, and I would say as a result of that, probably 50% of the sequences in Uniprot have incorrect annotations, but that's sort of the state of the art. We also are using the Uniref database. The Uniref database are clustered sets of sequences from Uniprot, where the one in particular that we use is Uniref 90, that sequences that share 90% sequence identity over 80% of their length are put in the same cluster. And the reason we do this is it reduces the number of sequences that we use in BLAST, because even though I've told you BLAST is fast, if you're doing BLAST for 200,000 sequences, that takes a long time, pairwise sequences. Okay, so, and why do we use Uniprot? It turns out that, and not NCBI. We use Uniprot and not NCBI because Uniprot, you can actually change an annotation. If you do an experiment, publish a paper, and determine that an annotation in Uniprot is wrong, they will correct it. You cannot do that with NCBI. NCBI is an archive. The annotations cannot be changed. And so it's for that reason that we use the Uniprot database. Okay, so we also use, for some of our options, we use the PFAM database, which is also associated with EMBL, and the Interpro family database, which is associated with EMBL EBI. PFAM is a database that is curated families, and there are now 16,712 PFAM families that are in the Uniprot database. I will say about a quarter of these are domains of unknown function. So even though they are curated as being in families, the functions are not known. Okay, and then Interpro has on the order of 34,000 families. This Interpro is the aggregation of 14 different databases, including PFAM. And so we make it easy with EFI-EST to access both of these types of families, both of these databases. Okay, so if you go to, so this is what the entry point for the web tool looks like. If you Google EFI-EST, the top entry on the list will be the link to these tools. And so this is the entry point, okay? And this entry point gives you access to both EFI-EST for generating sequence similarity networks and also EFI-GNT for generating genome neighborhood networks. So if you select EFI-EST, this is what the home page for the EFI-EST tool looks like. Now, what we would encourage you to do And those of you who have signed up for the tutorial, we encourage you to get an account on our system. And if you get an account on our system, you actually, there is a record of the previous jobs that you have run so that you can go back to previous jobs. We keep jobs, I think, what is it, Remy, is it 40 days now?
15 days.
15 days. Okay, I guess some of us are privileged and have 40 days. Okay. Because we have to pay for storage, you know, we have to worry about these things. And I will say, I don't know if there's anybody from NIH here, I do not have money explicitly for this project. EFI-ES, the Enzyme Function Initiative, was terminated 2 years ago. We now have a program project which uses these tools, but there is no money in the program project to support these tools, but somehow we managed to do it. And I will say that we make these available to the world, and literally they are used around the world, and NIH is not putting any money into it. All right, so, but we did buy a cluster before we, with the EFI. All right, so how did I get off onto that rant? I'm prone to rants. Excuse me?
Stay on target.
Yeah, okay. All right, so, so if you have an account, you can access your previous jobs. Okay, so we have 4 options that are available for generating sequence similarity networks. A, B, C, D. All right, what is option A? Option A is that you can provide a sequence, and then the tool will run a blast against the UniProt database and will collect the closest homologs, the closest hits to your input sequence. All right, that's option A. Option B is that you can specify a PFAM and or interpro family, and those sequences will be retrieved and you'll generate the network for the specified families. Option C is you can input a FASTA file, which is a list of sequences, and option D is you can put in a list of accession IDs. All right, so we have 4 different options. So here's option A. All you'd have to do is paste in a sequence and hit go, and the all-by-all blast is run, and eventually you will get an email that says it's done, or if you have your user page, it will change colors when the job has been finished. Okay, option B is somewhat is where you can use PFAM and or interpro families. Also, you can specify PFAM clans. I, of course, think about protein families. I've been interested in the evolution of function in protein families. So I thought the only reason anybody would use this tool would be to enter a protein family. I would say that maybe 1/3 of the number of jobs that are run are protein family jobs, maybe 1/3 are blasts, and the other 1/3 are distributed between FASTA and accession IDs, okay? So we have, this is option B, you can specify a protein family and you can hit go. Now, we've recently encountered the situation where our cluster, there are 360 processes on our cluster, and sometimes we're oversubscribed. All right, so we are now very sensitive to this, or have become sensitive to the size of the job that's been submitted. I actually just submitted an application to NIH, to GM, to enhance, to double the size of our cluster, and we'll see what will happen. You know, cross your fingers. All right. So if you select a PFAM family that has less than 25,000 sequences, you can just use all of those sequences, and the network will be generated. The all-by-all blast will be generated. And that's 15,547 PFAMs out of 16,712, so that's almost all of the PFAM families. Okay, if you have more than 25,000 sequences, which is 1,157 of the PFAM families, we require now, and this has happened within the last few weeks, that you use the UNIREF 90 sequences, the sequences where you're using the sequences, a seed sequence, a representative sequence, which is, let me back up. All right, a UNIREF cluster, what is it? 90% sequence identity. All sequences that in a family that are 90% sequence identical to one another, over 80% of the length, are in the same cluster. The longest sequence in that is used as the seed sequence, and we use that for the blast. The identities of the other sequences that are associated with that are going to be in your SSN, but we only do the blast for the 90% identical sequences. This causes anywhere from a 2-fold to maybe a 100-fold reduction in the number of sequences that have to be blasted, and that means it's a lot faster, which means it conserves computer resources. So if you specify more than 25,000 sequences, you'll get a warning that it's going to be, or a notice that you're going to be using Uniref. If you have more than 200,000 Uniref sequences, you can't do it. And it turns out that there are 8 PFAM families, and none of those are, I don't think any of them are enzymes. They are ABC transporters and this kind of thing. And so, but it's just, we don't have the resources. Our cluster is not big enough to do this. Okay, and now we also have just implemented, if you go back to the EST homepage, the access to pre-computed option B jobs. That is, we have already done the blasts for some PFAM families and interpro families. And we did it actually for this meeting because one of Marty's former students, Lauren, I don't remember her name, sent me a list of families, and we did the all-by-all blasts for all of those. And that also includes up here the radical SAMs, the P450s, and now if you want to do networks on these you only have to do the analysis step. You do not have to do the blast step because it's already been done for you. And again, we did that to be sort of defensive of computer time, so that we suddenly are not overwhelmed by everybody wanting to do the radical SAMs, all right? They've already been done. Okay, so option C, you enter a FASTA, either FASTA sequences or upload a file, and then you hit go. And actually what this option will also, so this is a FASTA formatted sequence. There's a header with a sequence. The blast will utilize the sequence. The FASTA header has an ID in it, a Uniprot ID. And if you click this little box right there, the program will read the ID and it will extract from Uniprot the bioinformatic information that becomes the node attributes that are in the sequence similarity network. So you can generate a sequence similarity network reading those IDs. We will also read NCBI IDs. The NCBI IDs, if you have a list of NCBI IDs, probably half of those will have a Uniprot equivalent. The NCBI database is larger than the Uniprot database. About half, as I said, of the NCBI IDs will have a Uniprot ID. And again, it will read these sequences, and if we can find a Uniprot equivalence, it will provide the node attributes. Option D, you can provide either a list of either Uniprot and or NCBI IDs, and it will run the sequence similarity network. Okay, so when your job is done, you can, when the blast is done, you'll get an output page, and what's important on the output page are 4 plots. These 4 plots, 2 of these are histograms. One is a length histogram, so this shows you the length heterogeneity of your protein family. The other one is an e-value histogram, and it shows you the distribution of the number of times a particular e-value was calculated in the sequence comparison. And I'm not going to go into this. This one is useful. Okay, because the length histogram will allow you to see if there are perhaps multiple domains, if your protein family has members that have multiple domains, NRPSs, PKSs. It'll also reveal fragments. It's very common that there'll be sequencing fragments, and so you will get short sequences that are associated with, that are present in your family. I don't think I'm going to say anything about that. So you also get 2 quartile plots. One of these is very, very important, and this is the percent identity quartile plot. So this is what this looks like. This is the percent identity is a function, essentially, we call it an alignment score. It's more or less the e-value. So what this says is as the e-value gets smaller, as the alignment score gets bigger, the percent identity increases. Okay, why are we interested in this? We're interested in this because in constructing a sequence similarity network, I told you that there would be a line drawn between pairs of sequences that share particular greater than a user-specified percent identity. All right, so you have to know what do you want your lower limit on identity to be, all right? So let's say we recommend that you choose 40% sequence identity to start out with, because that may separate functions, all right? So a 40% sequence identity corresponds to an alignment score on this, for this particular example, of 33, all right? So this 33 is what you're going to enter into one of the panels on this page. All right, so on the same page, this is where you put in the alignment score. So this is going to draw edges only if the percent identity is greater than 40% for this particular set of data. You can specify minimum and maximum length. You have to put in a name for your network that'll appear in Cytoscape, and then you hit go, all right? When that is done, you get a download page. It's got some statistics at the top. At the bottom, it has various files that you can download. Okay, you have one file that you can download. It's usually there, but not always, is for the full network. That means it's a network that has all of the sequences, all right? We also provide what we call our representative node networks. And representative node networks collect together sequences that share 100%, 90%, 80%, 70%, 60% sequence identity, all right? And as you can see, as you make these metanodes, as you collect sequences that share smaller and smaller sequence identities, the number of edges decrease. All right? This is important, as I'll say on the next slide or two, is because Cytoscape visualization software is limited by the number of edges that can be drawn. And so the number of edges that can be drawn depends upon the RAM that's available on your computer. And so you have to download, if you want to work with these, you've got to be able to download a network that you can open. And so this provides you with the ability to download networks where sequences have been grouped by different levels of sequence identity. Of course, as you go down to 80, 70, 60, 50, 40, the resolution of your sequence similarity network is going to decrease because you're combining sequences that are sharing these low levels of sequence identity. Okay, so these are representative node networks. A useful guide is that if you've got 8 gigabytes of RAM, you can open a network with about 500,000 edges. If you've got 16 gigabytes, 1 megabyte, 64 gigabytes, 5 million edges, 128 gigabytes, 10 million edges. Okay, I happen to have one of these in my office and one of these at home. I'm told that lots of people don't have those. I don't get anything from Apple, but here's a price list. And you know, it's not really if you're really going to get interested, use these. You know, you ought to get your PI to buy one of these machines that has, 64 gigabytes or so that you can do some big things on it, all right? Do some high resolution networks, all right? They're really not that expensive, and not everybody needs one. You know, I have 2, but not everybody needs one, all right? All right, so the visualization is done on Cytoscape. Cytoscape is a downloadable platform for visualizing SSNs, and this is what a network looks like using Cytoscape. The network comes with node attributes. That is, with each node, we provide lots of different kinds of bioinformatic information, phylogenetic information, other databases, all this kind of stuff. And we'll talk about this in the tutorials this afternoon. Okay, Cytoscape also has a control panel. And what the control panel does, it has 3 control panels. One of them allows you to choose, you can open multiple networks, so you can have, it allows you to choose networks. The select panel allows you to select nodes that have particular values of node attributes. And then the style panel allows you to change the properties, size, color, whatever, of these selected nodes. And I will say that Cytoscape is the hardest part of this, all right? And we will spend a lot of time in the tutorial dealing with the Cytoscape issues. Okay, so here are some bioinorganic superfamilies. This is this list that Lauren provided me, and I just wanted to, this really isn't bioinorganic, is it? Glyceradical, of course, there's a radical SAM that activates these to generate the glyceradical, but this is the glyceradical network. You can see that there are a number of different clusters. This is probably fractionated for isofunctionality. Each cluster has the same function. Here are the 7 functions that have been associated with the glycol radical enzyme family. And the point of this is, notice that there are a lot of gray clusters. The gray clusters do not have functions, and so these are probably members of the glycol radical enzyme family that are doing some amusing chemistry that is yet to be discovered. Okay, everybody is interested in radical SAM. Okay, so here is a radical SAM network. Okay, so this is Tyler Grove, who I think a number of you are familiar with, suggested that I use an alignment score of 120 that corresponds to about 50% sequence identity. And when you do that, there are 15,638 clusters in this network. A cluster has more than one node associated with it. Okay? I've colored in blue those clusters that have a Swiss prot function. All right, so at least some of these are probably heterogeneous, but I've colored the whole function. So there are some clusters that are colored, but notice all of the gray ones. There are lots of gray clusters which are unassigned functions. Squire recently wrote a letter for me that went along with this supplement request about supporting our application, and I think this is probably pertinent to this audience, that there are probably a lot of careers invested or represented in the gray clusters in this network. All right? So this allows you to identify them and work with them, hopefully. Okay, I want to say a little bit about the GNT tool. I'm going too slow here. So here's the genome neighborhood tool. What is this for? If you have high sequence identity between two proteins, you can probably, if their sequence identity is, let's say, 70%, you can probably transfer the function. If you have lower sequence identity, it's not so obvious you can transfer the function, but maybe if you have genome context, that is, do two homologs share the same genome context even though they have low sequence identity, maybe they have the same function. So this is what genome neighborhood networks are for. So the input for this is a sequence similarity network with clusters. What we do is we identify the members of these clusters in the genome databases. We retrieve the neighbors. We partition them into PFAMs, and then what we do is associate with each cluster the PFAMs that are close by. All right, and those PFAMs that are close by are probably or could be encoding metabolic pathways. All right. So this is accessible with the EFI GNT tool. We use sequences from EMBEL from the ENA database. And what you do is you input an SSN, you put in how big do you want your genome window to be, what do you want the co-occurrence frequency to be of your query in the SSN with your neighbors, and you put in your email address and off you go. Here's the input. You get back an SSN that is colored. Each cluster has a color. And then you get back another network that has, for each SSN cluster, it has the PFAM families that are in the neighborhood. And so what you can do is here for cluster 15, you can read off the enzyme families that are in the genome neighborhood. And if you're a chemist, and everybody in this audience is a chemist, you can probably use the PFAM curations to begin to infer a metabolic pathway. The PFAM curations will tell you the kind of chemistry, the kinds of reactions. Okay, we also provide another GNN format, and what this is, for each PFAM family that was identified, for each PFAM family that was in a genome neighborhood, we identify the clusters that found that. So if multiple clusters found the same PFAM family, it could very well be that these clusters have the same function. And the whole problem with these SSNs is how do you segregate them to be isofunctional, and so this should enable that. We also now have, for each cluster in an SSN, we make it very easy to look at the genome neighborhoods. This used to be very cumbersome to do, and now you can just simply, with one of the boxes, one of the output selectors, is you can say you want to look at genome neighborhoods, you put in the cluster number, and there they are. Okay? So this is now very easy. So you can examine the genome neighborhood for every cluster, every sequence in every cluster. Okay, so I want to spend a few minutes talking about the use of SSNs and GNNs to discover pathways. And I'm going to talk about solute binding proteins as a way to facilitate the discovery of new pathways. And these are solute binding proteins for transporters. Solute binding proteins, there are ABC and TRAP transporters. These have extracellular or paraplasmic solute binding proteins that will bind the solute that is going to be transported by the membrane components of the transporter. Okay, if you can purify these on large scale, and you can then do a large scale ligand screening of these, and so that you can take we have a library of about 500 compounds, and so we can screen solute binding proteins against 500 compounds. This is carbohydrate enriched because of my previous interest in the enolase superfamily. And so we can do this easily. Okay, so we did this for members of PFAM 13407. This is one of the solute binding protein PFAM families. And we discovered three solute binding proteins that had hits with apiose. What is apiose? I didn't know what apiose was. Apiose is a branched carbohydrate. Apiose happened to be in our library because it's an aldose that you can buy. Okay? And also, let me just point out, I think I passed over this. What I'm telling you about today will be available online in Nature Chemical Biology on Monday. All right, so this is, you can look at all the details on Monday with this. Okay, so these bound apiose, but they also bound ribose and ribulose. So we did the knockouts of the genes encoding the transporters, solute binding proteins, and also some pathway enzymes. And then we challenged these, or used apiose, ribose, and ribulose as potential carbon sources. And it turns out apiose was the one what was affected. So D-apiose was the carbon source, all right, is the solute binding protein ligand. Okay, what the hell is D-apiose? D-apiose turns out to be a component of rhamnogalacturonan 2, which is one of the plant cell wall polysaccharides. And it turns out, here's apiose. It turns out that rhamnogalacturonan 2 strands are connected are linked to one another by boronate esters. And those boronate esters involve the vicinal hydroxyl groups of apiose. All right, Bacteroides, a polysaccharide utilization locus in species of Bacteroides from the gut microbiome has been identified that completely degrades rhamnogalacturonan 2 to apiose. And so apiose is available, but there were no catabolic pathways known for apiose, so we thought we would figure out catabolic pathways for apiose using SSNs and GNNs. So this is the sequence similarity network for the solute binding protein family, and all three hits are in that cluster right there. All right, so these appear to be orthologs. This is an orthologous cluster of SBPs for apiose. Okay, so we put this into the genome neighborhood network tool, and when we did this, we got a whole bunch of enzyme families that were in the plus or minus 10 ORF window. All right. And if you try to draw a pathway for that, you'll say, there's far too many, I got too many enzymes. So if you increase the alignment score, that is, if you segregate that single cluster into two, you can then do a genome neighborhood network on that, and it's much cleaner. Okay? And it turns out that this is one pathway, and that is three pathways. All right? All right, so what is this pathway? This pathway has a transketolase, a kinase, and an isomerase. And so what we've determined is what the kinase does is it phosphorylates apiose to make apiose phosphate. The isomerase makes apulose phosphate. And then the transketolase uses glyceraldehyde 3-phosphate and apulose phosphate and makes D-xylulose 5-phosphate and dihydroxyacetone phosphate. So this is in the pentose phosphate pathway. This is in glycolysis. And so this is why apiose can be utilized as a carbon source. And this is actually, this pathway is found in a number of Bacteroides. All right? So we discovered a pathway. Now what about this other one? This other one has a bunch of dehydrogenases, and it also has members of the Rubisco superfamily. All right, so what are the dehydrogenases? So one of them oxidizes D-apiose to deapianolactone, and then there's a lactonase that opens this to deapionate, all right? All right, so there's another so that was this dehydrogenase. There's another dehydrogenase, and what that dehydrogenase does, and it uses NAD+, I don't have the cofactor here. What it does is it oxidizes apionate to this 2-oxoapionate, and then there is an intramolecular migration of a hydroxymethyl group to make this compound, which we call 3-oxoisoapionate, and this is a beta keto acid. Okay. We don't know what, so this is a novel enzyme. So I'm giving this as an example. We discovered a novel enzyme. We don't know the mechanism. We haven't figured out the mechanism. We don't know if this is a concerted reaction. We don't know if it's stepwise by generation of a formaldehyde intermediate. But if we had the time and interest, we could try to sort this out, all right? But this is a novel enzyme.
So there's no indication of a cofactor in the sequence?
Oh, this is, so this has it's an NAD, it's a 2-domain, it's a dehydrogenase, okay? And you have to, I left the NAD out. We have to provide the NAD. It does not have NAD bound, all right? But this is a novel reaction. Okay, so if you take this, and we call this an oxidoisomerase, this is the sequence similarity network for the oxidoisomerase superfamily, family. It's quite small. But at this alignment score, there are 2 clusters. One of the clusters has one genome neighborhood. The other has another kind of genome neighborhood. Okay, if we look at number 1, there is a kinase, there's a member of the triosisomerase family, there's a member of the ribose phosphate isomerase family, and of the xylose isomerase family. It turns out what those enzymes do, they take, here's 3-oxoisoapionate, which is the product of that oxidoisomerase. The xylose isomerase catalyzes an decarboxylation to erythrulose. The kinase phosphorylates that. Here is triose phosphate isomerase doing a 1,2 proton transfer on a 4-carbon substrate instead of a 3-carbon substrate. And here is this ribulose phosphate isomerase homologue. So this generates erythrose 4-phosphate. Erythrose 4-phosphate is in the pentose phosphate pathway, so that says why this pathway can be used for carbon source utilization. Okay, what about this other one, the other cluster? It's got Rubisco-like proteins in it, or members of the Rubisco superfamily, and I've been interested in those for a while. So what are these? So this is a sequence similarity network of the Rubisco superfamily. And this is a 95% rep node network, so sequences that share 95% sequence identity are collected together. Now, I want to just point out that it turns out Rubiscos are pretty identical to one another, okay? And remember I talked about this UniRef90 database that collected sequences that were 90% sequence identity into clusters. Turns out the Rubisco superfamily has 137,000 sequences, but it only has 5,000 UniRef90 clusters. If I were to do the all-by-all on 137,000 sequences, which I've done, it would take several days. On the 5,000 sequences takes 15 minutes. So this is an example of how UniRef90 can be used to speed things up. Okay, 3 of these are Rubiscos, authentic Rubiscos, Form 1, Form 2, Form 3 Rubiscos. The magenta one and the cyan one are other Rubisco-like proteins that I'm not going to tell you what they are. They're involved, we know what they are, they're involved in methionine salvage, but these involve enolate anion intermediates. This cluster is the cluster which is proximal to the oxidoisomerase, the cluster that's making this 3-oxoisoapionate, all right? So if we take that cluster and we go to a larger alignment score, that is a greater percent sequence identity required to draw edges, we get a bunch of clusters, but we get 2 major ones that are proximal to the oxidoisomerase, all right? So what are they? So this one, it turns out triose phosphate isomerase is close by, ribose phosphate isomerase close by, and there are a whole bunch of kinases. Well, I just told you about a pathway that has triose phosphate isomerase and has ribose isomerase homologues in it. And it turns out that what this pathway is, it takes 3-oxoisoapionate, it phosphorylates it to make 3-oxoisoapionate phosphate. Then the Rubisco-like protein catalyzes a decarboxylation. So Rubisco catalyzes a carboxylation. This catalyzes a decarboxylation. So Rubisco stabilizes an enolate anion that allows that to occur. And then here's the triose phosphate isomerase and ribose phosphate isomerase we saw before. So again, there is the formation of erythrose 4-phosphate. So what we discovered are two ways that 3-oxo-isoapionate can go to do deerythrose 4-phosphate. One was with the xylose isomerase, where the decarboxylation occurs before phosphorylation. And we've also discovered where phosphorylation precedes decarboxylation, and you end up with both pathways making erythrose 4-phosphate. All right, so there are two pathway variants. All right, probably the most amusing, as far as I'm concerned, is this other one, is this other cluster in the Rubisco-like proteins. And so it has a kinase, it has the oxidoisomerase 3-oxo-isoapionate, and it has the enzyme that oxidizes apiose to apionate. Okay, so what does that do? It turns out that it takes the phosphorylated 3-oxo-isoapionate, and this is decarboxylated, and then that carbon dioxide actually moves from one carbon to the adjacent carbon, and it carboxylates, so you generate an enolate anion intermediate, and then the other carbon of the enolate anion intermediate is carboxylated. And then this is hydrolyzed, and you make glycolate and 3-phosphoglycerate. I didn't draw because I didn't think I was going to have time, but it looks like I had more than enough time. Am I going so fast that you can't understand? Or are you following me? I could have put more slides in. So this is basically the Rubisco reaction. What do we have? We have the substrate brings in carbon dioxide, and makes, so here we have carbon dioxide. There's an enolate anion intermediate, which is what Rubisco would generate. And then that enolate anion intermediate is used to carboxylate. You form this ketone, which is then hydrolyzed, and that's just like the Rubisco reaction, except the substrate brought the carbon dioxide substrate in. So I think that's quite interesting. So what we found here are two different Rubisco-like proteins. One that does a decarboxylation reaction, the second does a decarboxylation but then uses that carbon dioxide to carboxylate the adjacent carbon, and then it's hydrolyzed, and you form 3-phosphoglycerate and glycolate, and both of these can be used in central metabolism, so that explains how this pathway can be used to provide growth on apiose. Okay? So what we did is we started with 3 solute binding proteins. All this stuff I just told you about. We started with 3 solute binding proteins. We used sequence similarity networks and genome neighborhood networks. We discovered 4 pathways, and those 4 pathways are present in greater than 1,300 bacterial species. So we only started with 3 solute binding proteins, 3 pieces of information, and we got 4 pathways in 1,300 organisms. And I think that's a fairly good return on the investment. All right? So it says how the sequence similarity networks and genome neighborhood networks can leverage pathway discovery. Okay. I want to say just a little bit about this shortbread, this chemically guided functional profiling. As I said, last February, Emily Balskis and Curtis Huttenauer published a paper in Science where they assigned functions to 2 different families of the glycerol enzyme family. One of the functions was 4-hydroxyproline dehydratase, and another one was a 1,2-propane-diol dehydratase. And what they did, so these were activities that are found in the gut metagenome. All right? And what Emily and Curtis described was using the abundance of members of protein families in metagenomes and mapping those to sequence similarity networks for the protein families to figure out which clusters, in their case, the glycol radical enzyme family, were represented in the human gut microbiome. And so if you're trying to identify a problem, an activity, an uncharacterized cluster that might have human health relevance, it might be useful if it would be present in, for example, in the human gut microbiome. And so this is the glycol radical enzyme family, SSN. I showed you this previously. And I said that there are 7 characterized functions, and here are the clusters, here I've colored the clusters with those known functions. And you can, as I've told you before, there are a lot of gray clusters here that correspond to uncharacterized functions. Well, these are the clusters that are represented in the human gut metagenome. Okay, so you can see that some of those went away, some of the characterized ones went away, but what you're left with is a lot of gray ones that have unknown functions. All right, so this chemically guided functional profiling allows one to identify these clusters that might be worth going after. And as I said, we have begun to do this. Actually, we have we have a beta tool that we have been using in Illinois. And, so I probably shouldn't say that we have this because we haven't quite made it public yet. All right. But it's pretty simple. We put in a sequence similarity network for a protein family. What happens is this software, this procedure, is shortbread that Curtis Huttenauer described. It determines unique sequence motifs in the clusters in the sequence similarity network, and then it uses those unique sequence motifs to probe the metagenomes, okay? So the first step, you input the SSN, you identify markers, and then we have a library of human microbiome project genomes that we can use as input. We can select the ones that we want to search through, and then we have an output. And that's how I constructed this. These are the clusters that are represented in actually the human gut metagenome. So the problem with this is computationally extremely intensive, and cross your fingers that NIH will give us more clusters and even more nodes, and even then I'm not quite sure how we're going to handle this because this is big time computing. All right? But so anyway, what have I told you about today? I've told you about sequence similarity networks and how to construct them. I've told you about genome neighborhood networks and how you can construct them using our tools. I gave you an example of using these two, SNNs and GNNs, to discover functions, and then I've given you an idea of how we might be able to implement this prioritization of clusters using CGFP. So I want to thank my collaborators. Nils Oberg is the programmer who has been working on this, and Remy, who is here, has been absolutely spectacular, both of them, in making these web tools much more user-friendly than they used to be. And here are other collaborators. And as I said, you can access these tools just by Googling EFI EST and EFI GNT. And I think for those of you who are doing the tutorials, we want to make sure that you have, as we've asked you to do, that you've downloaded some software before you come to the tutorials. So with that, I will close and take any questions you might have.